Good afternoon, everyone. I'm Sangeeta Dwivedi, Assistant Professor of Modern Institute of Pharmaceutical Sciences, Indore. Today, I'm going to explain how to do bacteriological estimation of water by most probable number method. Today's objective is to enumerate the number of bacteria present in the drinking water by MPN method. So most probable number or MPN analysis is a statistical method which is based on the random dispersion of microorganism per volume in a given sample of water. In this method, measured volume of water is added to a series of test tubes containing a liquid indicator growth medium. The media receiving one or more indicator bacteria show growth and a characteristic color change uh, analysis. So color change is absent in those receiving an inoculum of water without having indicator bacteria. MPN test is done by three type of testing. First is presumptive test. Second is confirmed test, and last is completed test. Presumptive test looks the presence of fecal coliform in the water sample by inoculating lactose broth with the water sample. Those tubes that shows presence of acid and gas are positive in results, and those with no acid and gas production are negative in results. Three sets of lactose broth are inoculated with varying dilutions of the sample. First test of 3 or 5 tubes inoculated with 10 ml of water sample. Second test of tubes inoculated with 1 ml of water sample. Third set of tubes inoculated with 0.1 ml of sample. The combination of positives in the three sets is used to figure out the MPN or in 100 ml of water using the table which is provided in next slide. The tubes that shows positive in the presumptive test should be confirmed to contain Escherichia coli. This is done in the confirmed test using the selective or differential medium EMB, that is especially for Escherichia coli growth. The completed test is done only where legal issues are involved, when the bacterial culture is then identified by a full complement of tests including gram stain to show that it is indeed Escherichia coli. For that, the requirements are like this: petri dishes, test tubes, sampling water, McConkey or lactose broth medium, EMB agar or Newton agar media, Durham's tubes, test tube stand, and water sample. So the procedure of presumptive test is first we have to prepare McConkey media of single and double strength in test tubes. Then take 3 sets of test tubes containing 5 tubes in each set, and one set with 10 ml of double strength and other two containing 10 ml of single strength. Using sterile pipettes, transfer 10 ml of water to each double strength broth tubes. Then transfer 1 ml of water sample to each 5 tubes of one set of single strength broth, and transfer 0.1 ml of water to 5 tubes of remaining last set of single strength broth tubes. Then incubate the tubes at 37 degrees centigrade for 24 hours. Then after incubation, observe the gas production in Durham's tubes and color change of the media. Record the number of positive results from each set and compare with the standard chart to give presumptive coliform count and in per 100 ml of water sample. For confirmed test, take the positive tubes from the presumptive test and using EMB in duplicate. Then incubate one plate at 37 degrees centigrade for 24 hours and another plate at 44.5 degrees centigrade for 24 hours. Look for typical colonies in the media. Blue, black with green metallic sheen colonies are of Escherichia coli in EMB agar media. For completed test, inoculate the colony in a tube of lactose broth with Durham's tubes. Subculture the colony on Newton agar plate. This subculture is considered optional. Incubate the broth culture at 37 degrees centigrade and 44.5 degrees centigrade and nutrient agar at 37 degrees centigrade. Examine for acid and gas production in lactose broth. The Newton agar is used for gram staining and for IMVIC test. This is a flow chart of presumptive test. This is a water sample. We need to took 10 ml of water sample in first test tube set, then 1 ml of sample in second test tube test, and then 0.1 ml of sample in third test tube test. Then we need to check out the production of gas in our tubes. If gas is produced, then the result is positive. If gas is absent is there, then the test is negative for Escherichia coli bacteria. These are the picture of test tubes. This is the sample A. This is the first set of test tube. This is second set of test tube, and this is third test set of test tube. It is sample B. This is test set. This is the set of 5 test tube containing 10 ml of water sample. It containing 1 ml of water sample, and it containing 0.1 ml of water sample. So thank you so much.